Cell.
To sell.
Yeah! Hey everyone, welcome back to Cells to Cell. My name is Heem, that's Nick, and we're back at it for another Trivia Tuesday.
Yeah, let's do it. Today we have cell communication signaling quiz. So it's just a short 10-question quiz, but, to start off the year, I guess, for like this first month and a half or so, we're just going back to kind of basics of biology. Originally our goal for this podcast was to be able to bring science to people who might not be, in the field or even interested in the field. So we're just trying to continue to bring kind of light quizzes and, just fun ones for us that jog our memory and honestly take us back to undergrad.
So cell communication and signaling reminds me of probably cytokines, some sort of protein what is it called? Cascade effects.
Yeah, it reminds me of like the endocrine system, which everybody's kind of familiar with, but there are different ways that the body communicates, and the endocrine system kind of like is like the slow release. But yeah, that's just one way our brain communicates with our body, and vice versa. So
you want me to go first? You go first?
I don't care.
I say you go first.
Okay, I'll go first.
Okay.
Oh, this first question is like fun. All right. Hello, everyone. So I'll take the first 5. He will take the second 5. All right. Question number 1. Which of the following domain is not found in the structure of nuclear receptors?
Oh God, this is
not light.
This is not
light. Okay, so which of the following domain is not found in the structure of nuclear receptors? So, is it the hinge domain, the C-terminal domain, the DNA binding domain, or the F domain?
We haven't had a quiz like this in the ages, dude. This is hard.
This reminds me of like the first stem cell quiz we took on like WebMD or something. It was like a 5-question quiz, and it was just like impossible. So which of the following domain is not found in the structure of nuclear receptors? Is not found. I'm going to go with the hinge domain. Just sounds so cool. Like, I know that there was some scientists who found this structure. It was like, that looks like a hinge. That looks really cool. So I know that the C-terminal domain exists. DNA binding domain makes sense. So I'm going to go with the F domain because I don't... Because there's a C-terminus and an N-terminus, but that's mostly for proteins. But I'm going to go with F domain.
I'd go with DNA.
F domain's correct.
Oh, F domain's correct.
F domain's correct. All right, off to a good start. It was an intimidating start, but maybe the next question will be easy.
Oh, God.
Oh, this is not bad. What are the 3 stages of cell signaling? So this is a kind of a multi-answer question. So is it reception? So reception, transduction, transcription, response, translation.
Whoa.
So I'm going to go with, so this is cell signaling. This is not transcription, translation, protein synthesis. So this is not that. So I'm going to go with reception, response, and transduction. That's correct.
Nice.
That's correct. Cool. This is a little out of my comfort zone, I'm not going to lie. This takes me back to like molecular genetics, and I haven't taken a genetics course in a while. So, question number 3. Which of the following is an example of a second messenger? So, this is going to be something that, so if you think about a cell, something's going to attach to a surface protein or even maybe something inside. So that would be the first messenger. So that'd be the primary messenger. The second messenger is going to be what's going to continue to carry on that signal inside or outside of the cell. So is it CAMP, GTP, epinephrine, or insulin? So epinephrine and insulin are going to be primary. GTP is a maybe, but I think it's CAMP.
I would say so.
I think it's CAMP, or I think that's supposed to be a lowercase c, but I think it's C.
AMP.
Yeah, cyclic AMP. Boom.
Got
it correct.
Let's go.
All right. What are receptors? This is kind of okay. So are they catalysts, enzymes, acids, or proteins? So it's not catalyst or acid. And I don't believe it's an enzyme because technically the definition of enzymes is that they bring down the activation energy required for a chemical reaction to occur. So I'm going to go with proteins because when we think about surface receptors, they're going to be a lot of proteins or glycoproteins. And that's correct.
That was really good.
Was it?
That was good.
This is actually a good quiz. This is challenging. This is good. Okay. So my last question, question 5. Which signaling molecules do simple nerve reflexes use? So I think when you hear it, it's going to be a little more apparent because I've read through it and it seems pretty easy. So is it G proteins, nitric oxides, neurotransmitters, or none of the above? And it's going to be neurotransmitters. That's correct. So, yeah, neurotransmitters are really cool. Just to even study neurotransmitters would be awesome. All right, Heem.
All right, Nick. Oh!
This is a great quiz so far.
Let's see how I do. Question number 6. Which one is not a type of signaling molecule? Thyroxine, testosterone, insulin, adenylate cyclase. Which is not a type of signaling molecule. Okay. Testosterone is a signaling molecule. Insulin is I don't know if insulin is signaling one. Thyroxine and adenylate cyclase. I'm gonna say thyroxine. Ooh, the answer is wrong. Adenylate cyclase is not a type of signaling molecule.
What are the last 3 letters of that word?
ACE. Cyclase.
What's the X? What usually ends in ACE?
Proteins, enzymes. Yeah. All right. Question number 7.
Bounce back. We'll bounce back hard.
Yeah. Which is not the extracellular signaling molecule? Hormones, cytokines, neurotransmitters, serine kinases. Extracellular, so serine kinases. Hormones, cytokines, and neurotransmitters are all cell-to-cell outside. Serine kinases is like an intracellular thing, so it happens within the cell. 8. Question 8. Which is the highly conserved domain in the structure of the nuclear receptors? DNA binding domain, ligand binding domain, C-terminal, N-terminal? I know this.
What was the question?
Which is the highly conserved domain in the structure of nuclear receptors? DNA binding domain, ligand binding domain? I want to say DNA binding domain. The ligand binding domain should not stay conserved. C-terminal and N-terminal could also change. So, DNA binding domain is correct. Question 9. Which is not the function of the G protein coupled receptor? Glycogen synthesis and breakdown, regulates muscle contraction, inhibits aggregation of blood platelets, doesn't inhibit segregation of blood platelets. G protein coupled receptor. Oh my god.
This is a hard question. I don't know this one.
Like, do you remember G3PR? Like, these are part of glycolysis and thing. Ooh. I'm gonna say the function is not to regulate muscle contraction. Ooh. The answer is it doesn't inhibit segregation of blood platelets. So that's not a function of G protein coupled receptor. So it does inhibit segregation. So it stops
Cuts it.
the segregation.
Oh, yeah.
All right. Question 10, last one. Which is not the receptor of tyrosine kinase? PDGF, prolactin, EGF1, FGF. Receptor of tyrosine kinase. PGDF, PDGF. No, it's prolactin. Okay, that end was hard.
That was the last 5 questions.
Last 5. Not last 5, but last couple were definitely...
How do we get a score of 20 out of 100? That's wrong. I got 5 right.
And I didn't get all of mine wrong.
Oh well. Oh well. Hey, sometimes it's good to challenge yourself. We've said that before. So yeah, that was a tough one. That was kind of exhausting.
A little demoralized.
Don't be.
Oh, okay, it's okay.
Dude, you're not like. I feel like as you, I don't know, as you become more focused in like one thing, it's really hard to stay focused and remember everything else. Like,
yeah.
Like, how much World War II history do you remember from high school?
Dude, no.
Exactly, because you don't study that anymore. You are all science. So like your brain is just like science. We can throw all of the. You know, we have to remember a little bit, like, Nuremberg laws and everything, and, like, that's where we got our ethics from. But, you know, that's hard. Like, that was hard. That last question.
It's all specific.
No idea.
It's all specific.
Yeah. So, I'm not going to lie. I read the first 5 questions before I chose that. I didn't, like, read them. I didn't, like, read them and answer them. I just was like, okay, like, this sounds good. And I was like, oh, first 5 are good? Okay. So... Next time I'll be a little more diligent, but
no worries.
All right, you got anything else? All right, well, we just want to say thank you guys so much for watching. We want to say thank you for all the support you've been giving us. I know that a lot of, if you made it this far, thank you. But I know that a lot of the people we've had on the podcast, all of the guests that we've had on Fridays, have been doing really well. They get the word out, and we really, really appreciate that. So this has been Cells to Sell. It's been another Trivia Tuesday. And we appreciate you guys watching. I'm Nick and that's Heem. We'll see you guys next time.
See you guys.